Thanks everyone for joining. This is the last of the PFAS COP talks in our series, and we're very grateful to all the speakers that have participated. We have two wonderful speakers lined up for today, Wendy Linck from the California State Water Resources Board and Clare Pace from our own Community Engagement Center in our Superfund Center. Wendy is up first, and she's going to talk about the community water system sampling project. Thanks, Wendy.
Yeah, good. I guess, yeah, good afternoon, everybody. Thank you for the invitation. I'm going to include a little bit of water quality as well as the drinking water side, try to keep it high level. I do have a couple graphs that I'm going to show. So hopefully we'll get through this in good time, and then Claire can take the, I'll get to second base. She'll take us to home. And then I think we're going to take questions at the end. Okay, so I'm going to start off. So I actually sit in the Division of Water Quality, but we have been working with the Division of Drinking Water for many years now. I can't believe how long we've been working on PFAS now. We started in 2018 and now it's 2025. So I'm going to go over a little bit on our broad spectrum approach that we've been using on the drinking water side, a little bit on the investigation summaries that both have to do with water quality and drinking water, and in particular that disadvantaged community project that is currently underway. So for our broad spectrum approach, we're continuing to use this iceberg analogy. We're using, targeted methods for drinking water and for non-drinking water. But we're also have been working through and have been testing drinking water using non-target analysis, as well as absorbable organic fluorine modified method using EPA method 1621. So we are really trying to understand that entire iceberg with drinking water and moving into what we learned from there into the actual groundwater side as well. Essentially, drinking water is groundwater, right? But it is sometimes easier to work in these methods with a cleaner set of water than when you start dealing with wastewater and other complicated matrices. So we have been investigating. This is a map that shows we have issued through investigative orders to understand the presence or absence of PFAS across the state. We have over 3,000 investigative orders that have been issued. They have been targeting those industries that we knew that would have PFAS. Airports, for example, for the use of their aqueous film forming foam, and as well as refineries and bulk fuel terminals, the chrome plating industry for the mist suppressant. Landfills have to do with the leachate pretty much. And we also did an analysis for wastewater treatment plants. In tandem with that, we've been analyzing public water systems within 1 mile or 2 miles of those sources. And drinking water has been issuing their own investigative orders for public water systems to sample for PFAS and have been expanding outwards based upon detects. So this is kind of gives you an idea. We're pretty much covering the entire state right now with understanding the presence or absence of PFAS. I have one slide that kind of shows kind of the level in regards to water quality side, not the drinking water side. Essentially, this is kind of gives you an idea. And this, I have to admit, is over a year old data, but it's probably pretty much the same today. And essentially what it's showing you is that we have these different bins. The highest concentrations are in green. We see those compounds that are detected in PFAS in the highest concentrations at airports in groundwater and at terminals and refineries groundwater because of the use of aqueous film forming foam. So we have the greatest ranges of concentrations and impacts in regards to associated with those two industries. After that, we move to the chrome platers. We have a few instances where the chrome plating industry have some very large concentrations. But overall, we're down into the less than 100 nanograms per liter of any PFAS that's reported. And very similar to landfills, the POTWs, effluent, or even lower than that, we have quite a bit of lower concentrations having to do with wastewater. And then you can see the public water systems as well as of, and the number of samples as of a year ago. So we're going to switch to drinking water. Drinking water has issued essentially several orders, but there are two orders that are currently active right now. The one that was issued in 2022 and includes about 1,300 wells that incorporate about 400 water systems. These public water systems are required to sample quarterly based upon near to known or suspected sources. So back again, we're talking those airports, refineries, bulk terminals, chrome platers, and any of those secondary receivers of PFAS, which are landfills and wastewater treatment plants. In 2024, a separate investigative order was issued. And this has to do with the funding, a one-time sampling in regards to general funds that were allocated for the Division of Drinking Water to understand the presence or absence of PFAS in disadvantaged communities, those public water systems located. That includes currently right now, the initial intent was about 4,000 wells. We're currently running at about 3,600 wells. Several wells were dropped off the list because they're either not working or inoperable, which includes about 1,200 water systems. And I said it's one-time funding. The state is actually paying for this. It's not being, any costs are not being put onto the water system. We're currently enacting that broad spectrum approach. Every sample is being analyzed for EPA method 533, which is a list of 25 analytes, as well as AOF, the absorbable organic fluorine, the modified 1621. A subset of those, about 20% of the wells, because of budgetary costs, are being also analyzed for ultra short PFAS. There's about 6 of those. We're talking about those C2 and C3 PFAS. And we're also doing non-target analysis on that subset. We started in 2024 early, and we anticipate and are on schedule to end next year. So that funding that was provided to do this sampling project, we had to do several things. One, we had to select a broad spectrum method, and we went through a large study, and we went through and collected samples at 9 different wells, and we analyzed and sent them in triplicate. We did EOF, we did AOF, we actually did PIGGY, we did a whole bunch of different variations. And we wound it up that AOF was the better of all of them. It wasn't perfect, but it did provide the broadest chemical space. It did not provide very good coverage for ultra shorts. And that is why we supplemented our analyses to doing ultra shorts in regards to that. So the initial sampling project, as I said, was going to sample all these wells across the state. They cover from one end to the other. I'll have a map that shows those locations. We're running the 533 and we're doing AOF and the subset for ultra short PFAS non-target. So this is a map of those wells. This is a kind of a look on where we have sampled and we plan yet to go. And you can see that they literally are from one corner to the state to the other. There's a lot of density within down the Central Valley, but all along the coastline and over on the Nevada, Oregon, and borders. We're currently over 50% done right now. We have about 2,100 samples that have been collected. We've touched 500 water systems so far. And we're currently running at about 170 wells a month being sampled. We are going to maybe hit 185 to 190 during the summer. and we're touching about 40 water systems in doing so. Some of these water systems are very small, maybe one well, to other water systems that have over nearly 200 wells. I am going to show a couple charts in regards to just those results in comparison. So consider that statewide, the 2022 order, where we're kind of targeting, where we're kind of biased, where those samples were being collected of public water systems of known where there was going to be PFAS, airports, landfills, etc., versus the statewide DAC sampling. And so this is a chart showing the difference between the two, between the percent detects. And you can see quite a bit of difference between what was targeted, right? We have a running around 40% detect for those carboxylates and some of those sulfonates, and much lower detects, even 10% or 15% or less on detection for at the DACs, which makes sense. The other thing is that we're continuing not to see any of those other newer chemistries, none of the ethers, very, very few fluorotelomers in drinking water in the state, even when we are at the targeted locations or at the disadvantaged communities. We're pretty much in regards to what we're seeing in regards to method 533 is still holding tight in regards to those known carboxylates and sulfonates. So if you want to look at concentration ranges, these are the carboxylates. And once again, comparing between the two, between the kind of the targeted in blue and our DACs are in orange, you can see pretty much the range of concentrations without the outliers. We are running, below 20 parts per trillion, essentially, on the grouping of the main 50% of the data within that. And within the DACs themselves in the orange, if you were to look at PFOA, currently at the targeted, our median concentrations at 6.4, which would exceed currently the federal MCL for PFOA is 4, but the DACs, our median is about 3. So we do get quite a bit of non-detects in the DAC sampling, which we expected. We see a lot of great ranges in regards to our carboxylates. Within the sulfonates, even lower concentrations in regards to that main 50% within those box plots, even probably 10 nanograms or even 15 nanograms or less. PFAS is our leader here on the sulfonates. As you can see, our range is much more diverse for PFOS within the targeted, and that really has to do with those AFFF and those sulfonate-based products that we are seeing in drinking water. Our median concentration is 8.3, but the DACs, we currently are running a little bit high over the MCL at 4.6 for those. All right, this is just, I am almost done here, Claire. I've got 2 more slides to go. This is a map showing kind of the overall detection rate for PFAS, PFOA, and PFHXS based upon what were the current MCLs of 4, 4, and 10. And so pretty much we are seeing PFOA and PFAS and PFHXS. We see very little PFHX only. We will see PFHX with PFOA or PFAS or blah, 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 but very little water systems are being led by PFHXS currently. And last but not least, we currently have all this data in regards to the 533 data, and the AOF data is currently on our GeoTracker PFAS map. It is available for folks to download. I'm more than happy to help regards if you need any assistance in regards to that. So I just wanted to give a thank you, and I couldn't be here without my cohort, Dan Newton, in the Division of Drinking Water. This is a collaborative effort between both divisions as far as we've gotten so far. So we would be glad to take questions, I think, once Claire gets done. so let me stop sharing. And I'll figure out how to do that. Did I stop sharing, Claire? Are you up?
I think you need to actually end share
Right.
so that I can share my screen.
Yeah,
There.
try to do
Here we- Looks like I can send a request, but I can't actually share yet.
that. Yeah,
I think Wendy, is it in the share button at the bottom of the
it
screen?
is not. It's only allowing me to share.
It's only in oh, okay. Let's see, maybe I can stop.
You probably see my monitor screen now.
That's strange. I haven't seen this
I
before.
know it is.
I haven't seen this happen before.
I haven't seen this happen either. So what I'll do is I'll leave and come back in. I think that'll take care of it. All right.
Wow, that's strange.
Okay, I am able to share now.
Good.
So Wendy's not here, and I was going to thank her, but she gave a great presentation. I'm Clare Pace. I'm a project scientist at UC Berkeley, and I'm going to just briefly share some of the spatial data that we've developed to estimate sources of PFAS from agriculture and some of the preliminary results from our regional PFAS sampling study. So I'm presenting on behalf of the Water Equity Science Shop, or WESS. We're a community academic collaboration between UC Berkeley, UCLA, Kelly BA's Office of Environmental Health Hazard Assessment, and Community Water Center. And our work supports equitable implementation of California's human right to water. So PFAS are turning up everywhere we look, even in rural agricultural areas that are far from industrial sources. And this may be related to agricultural practices around pesticides and biosolids. So PFAS end up in biosolids because PFAS are present in household and industrial waste. Waste goes to treatment facilities and the PFAS don't get removed. Instead, long chain PFAS become concentrated in biosolids, and those biosolids are either applied directly to land as a soil amendment or they are transferred to landfills. So according to the National Biosolids Data Project, in 2018, California produced 675,000 dry metric tons of biosolids. About 66% of these were applied to agricultural land, and another 13% were disposed of in landfills. But no efforts had been made to map this. So we took this on, and we tracked down permits issued by the State Water Board to land apply biosolids as a soil fertilizer. So California has been tracking this to some extent since 1995. And this required us to go through a lot of these forms individually and find location data. It was a little tedious, but we were able to map 67 sites in California that applied biosolids and 100 waste facilities that accept biosolids. So another source of agricultural PFAS are pesticides. 66 active ingredients that are routinely added to pesticides are classified as PFAS. So we calculated and mapped the locations where any of these PFAS pesticides were applied in California between 2019 and 2021. And altogether, 8.5 million pounds of PFAS pesticides were applied in those 3 years. We also estimated how much PFAS pesticide was applied near public supply wells that serve community water systems. In order to do it, we created 1 kilometer buffers around the public supply wells, and then we used aerial apportionment, and we calculated the PFAS applied within that buffered area. Then we assigned that estimate to the service area boundaries for the water systems, and we calculated sociodemographics using American Community Survey data. And we published a paper in ES&T Water on environmental justice at the water system level with respect to these PFAS pesticides. Okay, so now I'm going to shift gears and tell you about how these data layers relate to our PFAS water sampling study. So we're testing household water from domestic wells and small water systems for 70 PFAS. This study is a collaboration with Community Water Center, and they're leading our sampling efforts in rural agricultural communities in the Central Coast and in the Central Valley. The sampling is focused in low-income communities that have a long history of water contamination. And we're using those agricultural PFAS layers I just described to inform our sampling strategy. So we're currently in our second round of sampling, and we're aiming to reach a total of between 40 and 50 households by the end of June. And I'm going to just show you some of our preliminary results. So on the left, I'm showing an example of a graph that a participant would receive as part of their individual results. The orange dot is the concentration detected in the household water sample. The blue dots are other detections from the region for comparison, and the gray dots show other non-detects. If there's a state or a federal MCL, that would also appear on the graph. And this report back is a collaboration with Silent Spring Institute. So on the right, I'm showing results from the first 22 households that we sampled. Altogether, we detected over 30 PFAS. The concentrations were higher and more frequent in households served by domestic wells compared to water systems. And this specific figure shows, I believe, 12 wells and 10 systems. Okay, now on the left, I'm showing the average concentration of each PFAS species that we detected. The error bars are standard deviation, and the number above the bar is the detection frequency in the well, which is in red, and for systems in black. So for example, 2 domestic wells samples contained PFOA, and the average concentration was around 30 nanograms per liter. 5 domestic well samples contained PFBS with a much lower average concentration, around 7.5 nanograms per liter. And so far, only FOSA was detected more in water system samples than well samples. Okay, and then on the right here, I'm showing the same data summarized by household rather than by PFAS species. So each dot on the map represents the total sum of all PFAS detected in each sample displayed in nanograms per liter. So for example, the largest red dot represents a household where we detected 10 out of 70 different PFAS, and the sum of all 10 was over 500 nanograms per liter. So finally, here I'm just showing some of the other contaminant data. We tested domestic wells for arsenic nitrate, 1,2,3-TCP, and hexavalent chromium. And to end on a positive note, based on the results I've shared today, plus results of bacteria and E. coli, we've been able to enroll over 20 households in state-funded bottled water programs. So going forward, we're hoping to wrap up sampling in the next month. We'll be analyzing spatial data trends. We'll report cumulative results to the communities where we've been sampling, and we'll be working with Community Water Center on strategies for policy advocacy. And I do want to reiterate that this study data is preliminary, so stay tuned. And I will end there and give us just 5 minutes for questions. Which, of course, feel free to ask questions of Wendy as well. Thanks.
Thank you, Claire. Thank you, Wendy. Rachel has a question. I'm not sure if she is still on.
Yeah, I'm still on. Thanks. Yeah, I was just curious, in light of mounting concern about biosolids, the extent to which the water board is thinking about using that as another criteria for targeted sampling.
Yeah, when we had our initial order, we didn't have any concentration that exceeded anything that would be considered elevated, Rachel. They were all below the EPA RSLs at that point in time. Right now it's funding, right? As we're funding and resource strapped right now. We are very fortunate to get the money that we have right now in regards to drinking water and doing this huge study for DACs. But we realize that there's definitely a different set of PFAS analytes we're going to see in biosolids and drinking water. So we hope that we can get there in the near future, but I'm having no.
Thanks.
Thanks, Wendy. Kathleen has a question. Kathleen, did you have a question?
I'm sorry, speaking while muted. Wendy, I was wondering if you could tell us a little bit about the AOF results.
Yeah, the AOF results are we are seeing about 30% detection for AOF. Currently, right now, we are seeing areas where we could have AOF reported and not anything on 533 or the reverse. And one of the reasons that is probably because of the detection limit. So we are currently have a reporting limit around 700 nanograms per liter. It can be, reporting limit, we can detect down about 200, but there is a gap there between 200 and where we can see method 533 down to 2 nanograms per liter. So what's happening is that we're getting non-detects for AOF because we can't see down far enough because drinking water concentrations aren't high enough. So that's preliminary. That's where we are in regards to AOF.
Thanks,
So
Wendy.
yeah.
Sorry, Narissa has a question.
Hi, thanks both of you. That was really great. Claire, I wonder if you could talk about the agricultural lands where there's been land application and what those lands are used for. Were you able to get that information from the permits about what happens to the crops? Is it grazing land or is it what's being done with the crops from that?
Yeah, a
those
lot
land
of that,
application
a
sites?
lot of that is agricultural crops, and so about a third of the permits list some sort of information on what crops are being grown. What might be more useful is using information from the Farmland Mapping Monitoring Service to sort of look at which crops have been grown in those locations over time, since it may not be the same as it was at the time of permitting. And really, the permits themselves are, they're not the most comprehensive data set.
Yeah, got it. Is there any thought about going to some of the land and doing any kind of core samples to see how the soil's impacted?
Absolutely. Yeah, that's exactly what I'd like to do next, is do look at sort of current and historical soil samples and sort of track trends in PFAS in areas where either biosolids or pesticides have been applied over time.
Great, thank you.
And Claire, when you move in that direction, we'd be more than happy to help collaborate if you're interested. It's something that we have been wanting to do and just haven't been able to move that forward. So we'd be more than happy to help support anyway, at least knowledge-wise, whatever.
Absolutely. Thanks.
Sounds good.
Much appreciated.
We have 2 more questions in the chat if you guys still have time. Wendy, I guess this one is for you from Amanda Palumbo. If PFAS are detected in a drinking water system, does the provider have to report it in the consumer confidence report?
Yes, they do. So there was a legislation that was passed back in 2019, 2020, and I might have gotten that year a little bit off, that required any detection be reported. So, and so you should be checking to see if your water systems are reporting any detection. They don't have to take action above these advisory levels that are issued by the Division of Drinking Water. So that's a little bit different, but yes, they are supposed to be reported, any detection of any analyte, not just PFOA and PFAS. So, yes.
Great question and interesting information. There we have another question in the chat. Has there been any effort to estimate background levels of PFAS in groundwater/municipal systems?
Not that I know of. There are several efforts that I know that are underway to kind of establish background in soil. And they're being either led by the military or by private consultants.
Yeah, and then the USGS also put out a PFAS prediction model that did some sampling, sort of random sampling, and tried to predict where shallow groundwater versus deeper groundwater may have PFAS concentrations. It's been kind of interesting to look at those results compared to what we're seeing in the shallow and deep groundwater in our sampling study so far.
Yeah, maybe Claire, sometime we can talk. It could be interesting to kind of line up those DAC wells with your locations, because I thought some of your box plots were very similar to ours.
Mm-hmm. Yeah, happy to talk further.
Yeah,
that's good. Yeah, I was wondering the same thing, how the areas lined up that you were focusing on.
Mm-hmm. Yeah,
I don't see any more questions. I think Junsu had his hand raised, but I don't see it raised currently.
Yeah, no, I think yeah, I'm gonna ask a question anyway because I missed the connection. You know, the area with the higher PFAS concentration, because your earlier slide were the biosolid. Were there any connection between the higher concentration over there with the biosolid application? Sorry, I missed the story.
Right. We don't have any water samples yet in areas that had high biosolid application. So, you know, that's one of the things we want to look at. I'd love to do additional water sampling in those areas. Yeah, that's a future analysis.
Thank you.
Mm-hmm.
It looks like there are no more questions. So I would like to thank Wendy and Claire for the fantastic talks. I'll share the recordings online and send out links as well to everybody in the audience. And thanks again to everyone for joining. Junsu, did you have a follow-up question, or your hand is raised, or?
No, this is not a raised question, just more like we collecting.
Okay. Martha has her hand raised if we have time for one more. Or she's clapping. No,
I'm clapping.
Yeah, yes.
Thank you.
All right. Okay. Thank you, everyone. Again, I'll share the recordings and the links. All right. Have a good rest of your day.
Thank you very much.
Thanks, everybody.